I'll be trying to give kind of connecting the dots and explaining how we both technically do this complex genome assembly and what's the value that we see in using this approach in kind of a broader look on the value that it can bring to different type of customers with different needs. So a lot of the talk will be centered around maize. And if you look at the original paper on the B73 maize genome, you see there was the sequence there, but even then they were talking about the complexity, diversity, and dynamics of that genome. So even in that original paper, they were focusing on the fact that in the maize genomes that there's a lot more than this just in this B73 genome. The maize genome is very diverse between the different lines, and to really appreciate and be able to work with a large collection and germplasm like breeding companies do, you need much more than one genome. And basically what we're trying to do is really enable that future of working with multiple reference genomes to kind of prove that, first of all, we can do that, and the value that we can actually deliver. We collaborated with two groups. One of them is Professor Mike Mickel from the University of Illinois, and we collaborated on the PH207, which is still an important line, and it contributes quite significant amount of genetic material into today's breeding programs. So we, with Professor Mikkel, we focused on that line, and we decided that having this iodent line will be a significant contribution to the community. And the other thing we did is with Professor Ed Buckler from Cornell, and as Faye mentioned in his talk, we sequenced and De-Novo assembled the CML247. You can see the details here. CIMMYT line, very representative of the tropical maize lines, and we were expecting to have that line very different from B73, and actually having PH207 more similar. And it is actually surprising that they're both very different. So this is the original data we got from the first two runs, around 120 and 110x. And as you saw in Faye's talk, the N50 was over 450,000 base pairs, and you can see the unfilled gap percentage, which is also a very important measure of the quality of the actual output you get. Okay, so if you want to be able to map things, and you want to see things that are, for example, the gene environment, that might be important if you're mapping, the percentage of unfilled gaps is becoming important. So what do you get when you're actually comparing the different genomes to one another? So Faye showed impressive numbers of almost 50% difference. What we get is about 60% homology between the genomes. We might be using a bit of a different parameters to decide what exactly is defined as identical, but we do get a major part which is significantly different. Still, the gene picture is a bit more complicated. So you'd see synthetic genes order pretty much kept. Of course, there are exceptions, like this is places that one of the two pairs kept the synteny and the other third member did not. And of course, the fact that there are totally new areas in one of the genomes, and they contain new genes. Now, for that analysis, we define the gene as long finished mRNA that was found in an experiment from a maize tissue, but was never aligned or not necessarily aligned to B73. So they're all proven full-length mRNA. Yesterday morning, Professor Candy Hirsch was showing a much more broad analysis she was basing on RNA-seq from PH207, and the numbers were much higher there. So there is a lot of new stuff to be discovered if you're actually taking mRNA data from the actual tissue of the reference genome you're sequencing, and not just basing your analysis on B73 or the historical data. So there's a lot to find. Now, how does that affect the actual gene content of the different genomes? So we compare the common and different genes for each genome. And again, I'm comparing this analysis to what Professor Candy here showed in her talk, and she found many more, like her numbers were in the thousands. So our analysis is quite restricted. And we're only talking about full-length mRNA found in a tissue, in multiple experiments in maize tissues. But still, there are hundreds of cases in which unique to CML or unique to PH207 and are totally missing from B73. So practically, if you are looking on, for example, QTL mapping, like people here showed that you've been done in for real research, you have a significant percentage of cases where you actually need to work with the right reference genome. And this is exactly the kind of vision we have. So we're trying to bring an era in which another reference genome will just be another common step in your research. People can do additional reference genome, they can compare reference genome to one another, and they can choose to work with the right reference genome, be it maize or wheat or any other complex genome that they might be working on. Again, the other important factor that you find is the copy number variation genes. So genes are not only duplicated, but they actually might be duplicated several times. And that, again, has a significant impact on the actual phenotype. And if you're mapping your QTL or phenotype to a specific gene, and this gene has 3 variants in that specific genome and you don't know about it, you might get to totally wrong conclusions about the effect of that gene. So what I'm trying to show here is kind of an example of exactly that impact. So when you're doing RNA-seq analysis, you're basically taking RNA-seq data from the tissue of a specific reference genome, in this case PH207. So again, Candy's Hirsch data, this RNA-seq data was aligned back to B73, like been done in most of the expression analysis data that all of us are doing, right? So there's this top hat approach. And just the default parameters, it's a very common tool. A lot of people do that, and they align back to B73. However, in this specific case, this gene here has 2 copies in PH207, and none of them really resembles the B73 original. So what you get from B73 alignment is a partial and quite hectic expression profile. And actually wrong one, because if you look on the right alignment of the RNA-seq same data to PH207 genome, you get a totally different profile, and it's split into 2 copies of the gene. Okay? So again, our vision is that you don't need to take this risk. You just you'll be able to do your own reference genome and work on the reference genome that's representative of your germplasm and your experiment. And overall site of the same data. So if looking on the PH207 RNA-seq data from several tissues and you're aligning that one to B73 and using the same software and same parameter, aligning it to PH207 assembly, you'd get a correlation of around 0.8. So there's a lot of cases in which there are 5-fold difference from what you get on B73 to what you get on PH207. And that's just one pair of genomes. Imagine the amount of differences you'll get if you're actually getting expression data from multiple genomes and you're aligning all back to B73. So you know, everybody destroyed my, my surprise here, but basically just hot from the oven results like Alvaro was talking about. When you're using this V4 chemistry and V2 rapid mode from Illumina, the amount of coverage that you can get for the same money and the amount of long reads using those 250 by 2 approach has really great impact on the N50 and on the unfilled gaps. So the assembly is improved in all parameters. And again, we kind of envision that as something that people would like to do a lot. This is commercial software. It's kind of should be in production mode and be producing more and more reference genome. And Maize is just one example. We're kind of looking at everything. We already have assembled polyploid genomes and long genomes, and we think this is kind of the way to go forward. So how do we do it? So I'm not going to go into the algorithmic details. The actual idea is that, okay, we have this de Bruijn graph like many other approaches have. The idea is to be able to kind of appreciate the heterozygosity or the similar to heterozygosity case in which things are duplicated and be able to kind of connect points along the bubbles created in the graph and kind of walk and create the phasing, the phased scaffold that are so important. If you try, a lot of the approaches either throw cases in which they're not sure what's the right path through the graph, or they try to kind of crush them down. A lot of the effort that went for Energene from the beginning was that our approach is that we need to do heterozygous genomes, and we built all our computational approach accordingly. So Faye already showed the same information. I'm just it's the same kind of objective test they did with the markers along their multiple markers along the Maize genomes. You would expect good context to be mapped to a single point on a single chromosome, and you can see the numbers. Most of our scaffolds are actually doing that, and this is information from the CML. I would say we didn't do that yet in W22 case, but we expect to actually be better now. So what's the value? How can this knowledge be translated into real life breeding or genetic variability data that people can use in real life? So what we envision is this kind of matrix or catalog in which this is just a visualization of the information behind the screens. And what we do is we color one genome, in this case B73, an arbitrary color like blue, and then we color the next one red wherever it's different from the first one. The next one is green wherever it's different from the first two, and so on and so forth. And pretty quickly you'd see lines that are actually a collection of haplotypes coming from other lines. However, in difference from the information Faye showed, we're not just using SNPs. So this is a total comparison of every sequence information that's behind the screens there. So it includes in deletions, insertions, structural variation, everything. What does that mean? It means that if you're focusing on a specific point, like this blue area here, in a specific line on a specific chromosome location, you can ask the system to give you primers that identify this blue region in all your germplasm collection. And they will be ultimate markers because they were compared to all the rest of the collection and be found to be unique to that specific haplotype in a specific position. So this becomes kind of a tool for real-life breeding or germplasm management applications that is very useful for people that are not geneticists or cannot have the bioinformatic infrastructure to actually do this for their own germplasm. Once we have that, they can just use it and have the value out of it. Another important factor for that is that if you can see this populations here, it's actually a derived line. So the lines that have their parents somewhere in the collection. And so you can see their genomes is basically a collection of 2 other genomes. And what's nice about it is now you can have 1,000 individuals, and you will know the exact sequence of each of those 1,000 individuals because you know their parents, and their parents are in the database. And this is quick, it's very applicative, and you can have that tomorrow with as many reference genomes as you would like. So this is commercial production-ready software versus just something that is used in research. So Faye also showed our ability to look at short reads and then map, and this Asaf also talked about it, the way we kind of map the scaffold over the genetic map. This is important mainly in cases where there's no reference genome. And so by using this genetic map ability, which is pretty unique to plants, you would be able to come up with a very accurate map for something that never had a reference genome. This is an offer for our customers in cases where they just want to get into genomics. But again, we think that nowadays they can jump into genomics in a stage whereas like the cutting edge of the crops that are already being worked on for a long time. So in a very short period of time, they can have reference genomes, they can have a lot of genomic variability computed, and they can have the system to manage the whole thing. And this is kind of describing the total approach. So we start with only short Illumina reads. If you go under this direction and you do a high coverage, you can get to, as I saw, 1.7 million and 50, and you can get all this nice things that you get from a reference genome. But this is expensive, and you would not do that for a long lot of lines. However, once you have several of those, you can go on the other path and have, by just 20x coverage, you can have a very good analysis of your haplotypes, and this is becoming a very useful tool for your breeding or your genetic management going forward. And of course, once you have the haplotypes, kind of understanding what's the polymorphism infrastructure or map that underlying the haplotype is very easy. So this is kind of real-life analysis of multiple groups, heterotic groups in today's maize germplasm. As you can see, there are multiple differences within the groups, but the differences between one group and another could be very, very impressive. And as we're looking on something like that, and we're imagining an organization trying to move from North America, for example, to Brazil or some other geography in which they need a totally different collection of germplasm, then we're imagining the use of this tool becoming very useful when you're trying to pick up things that are not too far away but not too similar to what you already have, so you'll be able to get the optimal breeding plan going forward. You don't need, if you're putting something that's too far away, you'll never be able to control it. If you're not you're not pushing it enough and you're just having something that's very unique, close by genetically, then you're missing a lot of this huge variability that's in there in maize. And I think what Faye was referring to in his talk, so having this huge collection of maize diversity inside will actually be going in that direction. So people in diverse geographies around the world will all be able to use this centralized database of variability and make the best decision, not based on what they just have, to happen to have in their collection, but really understanding the similarities and differences in the overall maize germplasm. So to actually offer that ability to our customers, we've developed a system. This is a very high-level view of it. The idea is that we're kind of having this huge database behind the scenes, in collecting all that very detailed information about very many different lines, for example, in maize or wheat or whatever your crop is. And then we have this the differentiating stakeholders in the organization. So molecular breeders, trait providers, bioinformaticians, each one needs different things from the data. And what we do is we supply kind of a user interface layer in which molecular breeders will get a very nice viewer that allow them to do things just by UI. And bioinformaticians can just program using our API and get the information they need just by writing their scripts. And then they can go on with that information to downstream bioinformatic analysis pipelines that already have been written. So we're kind of trying to help and not just replace the whole infrastructure they have. Of course, a lot of the people already have a lot of microarray data. And if you're used to your microarray and it's helpful, there's no reason to drop it. What we can do is to kind of integrate the data coming from the microarray with the data that we have and impute back the haplotypes. So just as a summary, basically what we're trying to do is to allow you to synchronize your old data with the cutting edge data, jump into genomics, even if you don't have a reference genome or if you thought it's too expensive. Now it's practically and doable and not that and pretty cost effective, and we can help, and we want to hear your case. So saying thank you to all the people that have been helpful in getting us to where we are. Of course, the PH207, of course, I mixed everything. So PH207 is Professor Mark Mikkel and Elvaro, of course, that was very helpful and very professional in actually producing the data. PH2 CML data was in collaboration with Professor Ed Buckler. The W22 data was done with a collaboration of researchers from different places. They're all very useful and very helpful in both understanding why is their line important and by delivering us the right amounts of DNA and so on. And the Energene team that here in the whole list of it, it's an effort by many people during a very long time to improve the system, both on the sequencing and the sequencing recipes, I would say, the right recipe. And here we really wanted to be able to do it cost effectively. It's very important for us to be accessible to the community, that people will not have to pay too much to get the reference genome, because what we envision is the ability to have many reference genomes in very short time, and we think that's the value. So the management and the ability to understand and compare genomes to one another is much more important than the actual cost of a single genome. And we hope that approaches like that will really get us to a point where there's no crop with no reference genome or with no genomic infrastructure, as there's no researcher that doesn't have an access to a system like that.
A couple of quick questions.
Sure.
I just listened to a talk by Ian McCallum at the Broad Institute on their Discovar assembler. Sounds like it has maybe a similar philosophy to your assembler. Just wondered if you had any comments on that, on how it might compare or contrast.
Actually, if you heard Faye's talk, he actually compared our results with Discovar. Discovar's actually approach is similar on the philosophy, and the technical details are different. And I think our is better attached to a specific recipe of Illumina reads, libraries, and coverages that giving us an advantage that Faye actually pointed to, on the overall N50 and the length of the contigs, which makes it just more practical to use and more cost effective.
Okay, I missed that talk, so,
okay.
So I'll look back for that. And then, do you have any comments on using this as in complement with long-read data from, say, PacBio?
Okay, so in the W22 case, we're already mapping, I think, around 95% of what we can scaffold, back into our pseudomolecules. So there is, of course, gain to be get from long reads, and the longer they will get, the more of the gaps that will be filled and order might be improved. We see our approach as a more cost-effective, large-scale thing. So you don't want to put so much effort and money into long reads if you can do another genome and another genome, and then the comparison will actually give you more than the filling of the exact gap. And it's as a use case by use case. Of course, if we are given PacBio data, it will improve the assembly. We're not recommending it from the cost-effectiveness point of view at this time, because you need to fill up their error rate. You'll need pretty high coverage. It's very costly, and it's also get more computationally challenging to put it on. And we see that for practical reason, usually what we get for the W22 will be more than enough.
Okay. Your pipeline can incorporate that if it was?
Yes.
Okay.
So thank you so much for coming, and I hope to see you in my booth.